Hey, everybody. So I know that you've all done Punnett squares, right? In middle school and high school, you've done Punnett squares and these probability charts and figured out crosses with capital and lowercase letters. The big question for today is where does that come from? Like, where did these probabilities come from on a cellular level? What processes lead to those? So, what we're going to do is we're going to look at a cell going through the processes of mitosis and meiosis and see how meiosis specifically explains what shows up on a Punnett square. And hopefully that will sort of connect the dots between cell biology and the mathematics of genetics. So let's go to the whiteboard. And on here, what I'm going to do is I'm going to just draw a simple model of a cell. And so I'll just big old oval, and within it, there's going to be a nucleus. And if this were a human cell, I think you know that there would be 46 chromosomes here. All right, so most of you have heard of that, 23 pairs of chromosomes. And the way you designate that is to say 2n equals the total number of chromosomes, 46. And the explanation here, n is the total number of types or sizes of chromosomes. We have 23 different chromosomes, designated chromosome 1, which is huge, chromosome 2, which is slightly smaller, chromosome 3, which is slightly smaller, et cetera, et cetera, et cetera. And so there are 23 pairs of these chromosomes of different sizes. On each chromosome, there are different genes. So this chromosome might include some like motor proteins. This chromosome might include DNA replication proteins and structural proteins. This may include the genes for growth factors or signaling molecules. In fact, it's even more complex than that. But this is the total number of chromosomes in a typical human cell. I can't draw that many chromosomes down here. So what I'm going to do is simplify this to a cell that's only going to have 3 sizes of chromosomes, 2n equals 6. If you're wondering, why are we talking 2 of everything? Well, remember, you're a diploid organism. That's what 2 means. And diploid means you got chromosomes from one parent and from the other parent equally. And so you have a chromosome 7 from your biological mom and a chromosome 7 from your biological dad. So the way I would draw this is, let's say this, again, this cell has 3 sizes of chromosomes. There's this one from your mom, this one from your biological mom, and this one from your biological mom. And then there's going to be a version of that that you got from your biological dad. So the long one, the medium one, and the small one. Okay, so that's our starting point. And this is a cell that we're going to say is, say, in G1. So it's entering into some type of cell division process. Now, as you all remember, after G1, if you've passed various checkpoints, then you're going to pass into the next stage, which is S phase. And during S phase, we're going to replicate these chromosomes. And this is where we're going to start talking about some of the vocab that you've heard of before, the really annoying and complex vocab, things like sister chromatids and homologous chromosomes and all that. So the cell should be a little bit bigger, but I'm not going to be hyper accurate here. But after S phase, we will have replicated each of these chromosomes. So this long chromosome that you got from your mom, it has been, this double strand represented by a single line, has been replicated. And there are now 2 identical copies right next to each other. Okay, same thing with this one. Has been replicated and has an identical copy right next to it, held together. This one, same thing. And I could do the same thing for the other versions of each chromosome. The one you say got from your dad, the long one, here's its copy, et cetera, et cetera. Please recall each of these lines represents a double helix. Okay, so that's after S phase. So now I suppose we are in G2. Okay, now what are we going to go into next? And what is the vocab of all of this? So one thing I'll let you know is there are these terms you've probably heard of, sister chromatids and homologous chromosomes, which get really confusing. Like, what's the difference and why are we using those terms? I would say first, chromatid versus chromosome, do not sweat that one. That is a waste of your time to worrying about chromatid versus chromosome. However, sister and homolog are the terms that I think are best to delineate. So let's just look at 2 of these chromosomes here. Let's look at the big ones. And so I'll just sort of redraw these larger. Okay. So there's this big one here, and it's, copy. And let's just say that there are, there are hundreds of genes on this chromosome, right? But let's say that here is where you have the genes for, let's say, primase, and maybe here is going to be helicase, and down here is keratin. That's where the genes are located. Remember, every person is going to have different versions of every gene. So we could say that this right here is P7, like primase allele 7. I'm not going to do capital or lowercase. There's so many alleles. And let's say it has the second allele for helicase. And for keratin, it has the fourth. Well, this other copy of chromosome 2, it's going to be exactly the same, because during S phase, we precisely replicated every single nucleotide of DNA. So when we copied this, we got the same allele for primase, the same allele for helicase, and the same allele for keratin, whatever those might be. So I'll just write those in, P7, H2, K4. All right, so what do we call these two chromosomes here? These two are identical, and the term for identical chromosomes, letter by letter, every single letter being exactly the same, and these two are attached as well, these two are known as sisters. And that means they are identical. And the full term that you've heard is sister chromatids. And the key word is these are genetically identical. They are the results of S phase. We went through that, copied it. Okay. What about this big orange one? It's the same length, right? It's the exact same length. It has the same genes. It has the same genes, meaning the orange one has a primase gene right here, and it has a helicase gene right here, and it has a keratin gene right here. This is the one you got from your mom, so I'll label that. Maybe this is chromosome 1 from mom. This is the version you got from your dad, right? Your biological dad. So chromosome number 1, paternal, as opposed to maternal, okay? The versions of each gene are going to be a little bit different, right? They might be, they might not be, all right? Let's say that the version that you got from your dad's chromosomes was primase version 11 and helicase allele 4. And your keratin allele that you got from your dad, maybe it was the same one as you got from your mom. But the chromosomes are not identical, right? This one you got from your dad, this one you got from your mom. They're both chromosome 1, same genes, same length, but not genetically identical. The term for that is homologous. So these are similar, but they are not identical. So this is where we switch to the other term, which is homologous chromosomes. So this versus this, homologous. And I'll just use the term homologs. Similar, but not identical. Okay, so there's our starting vocab, the stuff that people tend to get annoyed with. Okay. So if you need to pause there, you pause there, but I'm going to move on. So here we have a cell that's gone through S phase. It is now ready to go into some sort of cell division process. And the two processes we're going to talk about are mitosis and meiosis, and I'll delineate why we do each of those. And keep in mind that this cell has to be signaled to go into one or the other. So if you get, say, I don't know, a peptide growth factor signal, you activate a signal transduction pathway, it turns on some genes, makes some proteins. That makes you go into the mitosis pathway, which I'm going to draw over here in simplified form. And if you get a different hormone, a different peptide hormone, or maybe a steroid hormone, that activates a different pathway that sends you into the meiosis set of processes. So you have different proteins around, okay? So this is going to be meiosis happening on this side. Over here, this is going to be mitosis. big picture, why are you doing each? Why are you doing one versus the other? Well, the difference is mitosis is strictly for replication. And when I say replication, I mean identical replication. So at the end of mitosis, all you've done, this is the simple version of mitosis, you just take these chromosomes that have been replicated and you pull the two identical sister chromatids apart. That's it. That is it. We can get into all sorts of complicated terminology and go into phases. But really, all you're trying to do is you're starting with this cell, G1, gets replicated, all the chromosomes get replicated, and then you're just trying to regenerate this cell. So in the end, we're going to end up with two cells. And obviously, I'm drawing them smaller now. And these two cells are going to be genetically identical to what we started with. Exactly the same. How do we get that? As we've talked about before, we take these chromosomes, we line them up, each one, right? And obviously, I'm not going to draw this in some perfect manner because in reality, this would be in 3 dimensions and it would be kind of hard to do. And microtubules, with the help of a large number of motor proteins, which are monumentally cool to learn about, we'll end up pulling these identical copies into 2 different piles, 2 different locations. You form new nuclei around them, and you end up getting one of these big long blue ones, one of these medium blue ones, one of these medium little ones, et cetera, et cetera, et cetera. End of the story, you end up with 2 of the original. That is a reasonably simple process compared to what we're about to talk about on the meiosis side. The meiosis side is where all hell breaks loose, okay? Because we're going to do a very different set of processes, because on meiosis, we're not trying to generate identical copies. So remember, mitosis over here, signaled by growth factors and setting up those processes, we end up with 2 cells genetically identical to the starting one, right? So genetically identical to the parental cell. That's mitosis. Meiosis is going to be very different. The reason is we have to generate unique cells, and they better have half as many chromosomes. Okay, so you might be wondering, why are we cutting the chromosome number in half? Well, remember, we're going to put sperm and egg together, right? They're going to fuse together to form a zygote, right? So they're going to fuse together to form a zygote, and that's where you come from, right? You came from a single-celled zygote. Now, you can't just take 2 of these cells and fuse them together, because if you did, that would be 46 chromosomes, 46 chromosomes. You would have ended up with 92 chromosomes. And then if you tried to have kids with somebody else that was like you, your kids would then have 184 chromosomes, et cetera, et cetera, et cetera, et cetera. So now you can't do that. You can't just fuse cells together with all of their chromosomes. If you want to generate a new unique zygote, 2n equals 46, all right? You need to take a sperm or a male gamete that has 23, n equals 23, and an egg, n equals 23. Add those together, you get this. So meiosis is going to have to generate sperm or ova, eggs, gametes, that have 23 chromosomes. And more specifically, 1 of each chromosome size. 1 chromosome 1, 1 chromosome 2, 1 chromosome 3, 1 chromosome 4, et cetera, et cetera. Okay, so how do we do that? Here are going to be our 2 processes. Get myself a little bit of room here. The first thing we're going to do is, and by the way, I'm going to lie in this as I do every day. Make some massive lies here, but I'll fix them. I'll fix some of the lies at least eventually. All right, so during early meiosis, this is going to be meiosis 1, the first of 2 division steps. Right? During meiosis 1, what we're going to do is we are not going to separate sisters. That's what mitosis was. Mitosis mitosis, separation during, anaphase, that one step where you pull the chromosomes apart, okay? This is segregation of chromatids. So this is sister chromatid segregation happens during mitosis. That is not what happens during the first division of meiosis. During meiosis 1, you do this crazy process where you find the homologs and you pair them up. It's not just these sisters. It's not just these sisters. It's not just these sisters that line up. They are actually paired up with their homologs, like so. And which one goes on the left, which one goes on the right is randomized. We'll talk about that a little bit later. And so this is the lineup. And what you do during meiosis 1 is you don't segregate or separate the sister chromatids from each other. This is where you do segregation of homologous chromosomes. So homologous or homolog segregation is what occurs here. I'll, draw on a simplified meiotic spindle here. And it's going to pull this blue homolog this way and this orange homolog paired this way. This orange homolog, this orange version of chromosome 3 this way, this blue one this way. And yes, I've skipped a step. We'll get to that in a second. Please note, it didn't have to be this arrangement. Could have been. All the orange ones on the right. Could have, right? Could have been this. Who knows, right? It's all random. And we'll get to why that's important in a second. So in meiosis 1, you pull in this direction, separate the homologs from one another, and you end up with our first 2 cells. And this one over here is going to get, try to draw this appropriately, guess it was big blue, little blue on this side, medium blue on this one, big orange, little orange, medium orange. Okay, that was one possibility. Not the only possibility. There are actually lots of possibilities, right? Could have been all 3 orange on this side and all 3 blue here. Could have been, you can imagine the possibilities here. With 3 chromosomes, this cell could have gotten, there are 2 possibilities for the first chromosome, 2 possibilities for the second, 2 possibilities for the third. So there are actually 8 combinations of chromosomes this cell could have gotten. So this cell, which we'll eventually talk about what it leads to, there were 8 possible combinations of chromosomes it got. If you really want to blow your mind, or at least start to, remember that this is a stand-in for a cell that was had 46 chromosomes, 23 pairs. So if we separated 23 homolog pairs here, there would be 2 to the 23 combinations of chromosomes that you could produce in this cell here. And the same thing for this. All right. If you didn't follow that, not a big deal, but we'll continue. Right? Feel free to rewatch and rewind whenever possible, whenever necessary. Okay, so this step right here is pretty important because of the number of combinations you can generate. Therefore, this step has sort of a specific name, and the term is the English translation of something that came from Mendel, and this is called independent assortment. Right? The independent nature of this chromosome versus this chromosome versus this chromosome, the fact that this particular large chromosome, if big blue goes this way, it doesn't mean little blue has to go this way. The fact that this chromosome is independent of this chromosome is independent of this chromosome, you have all these combinations, is known as independent assortment. And that is one of the hallmarks of genetic diversity. That's why when you make your gametes, you can make, that's one of the many reasons why you can make so many, almost an infinite number of different eggs or sperm, depending on which one you make. Okay, so you might be thinking, all right, how many possible sperm could I make, or how many possible eggs could I make? Well, that's where the 2 to the 23rd comes from, which is a simplification, but that'd be about 8 million. So you might think, well, the possibility of like mom and dad having the same kid twice, and this is a simplification, the possibility of mom and dad having the same kid twice would be, well, you'd have to have the same sperm made twice and the same egg made twice. And that would be 8 million times 8 million, which means it's a 1 in 64 million chance of like the same two parents having the same kid. All right. Okay. That's if this was the only type of genetic diversity that happened. Let's stay with, let's just move on. We'll come back to that in a second. So this is one of the key steps of meiosis. This is one of the three, there's the four ways in which we get key genetic diversity. This is actually step 3 in ways of us getting genetic diversity. And we're going to backtrack the others. Okay, where else do you get genetic diversity? So this is one of them, getting different combinations of chromosomes. All right. S phase itself, DNA replication can have errors. So DNA replication errors, also known as mutations, can lead to genetic diversity. So what I'm listing here are the ways you can get genetic diversity in your offspring. Why do kids look different from their parents? Why does genetic diversity happen? One is mutations, and that happens during DNA replication typically. That's what we're going to focus on. There are other methods, but that's the one we'll focus on here. The second step I skipped because I wanted to make this drawable before we got too far. What's the second step? The second step is crossing over. So genetic recombination. So I'll simplify that. Genetic recombination, also known as crossover. And that is when portions of each of these chromosomes exchange with the chromosome next to it. And oftentimes people misdraw that. They draw this in some silly way where it's like, for some reason, they always draw things as X's, which is just silliness. So this is I'm drawing this just because I know some of you have seen this before. It's misleading. And people always draw it this way and they're like, they switched these little pieces right here. So this one gets the little orange end and this one gets the little blue end. Aren't they cute? They've like switched socks. No, that's not exactly how it works. Actually, there are going to be multiple locations here where they exchange material. And it's not just the ones that are in 3 dimensions, all 4 of these can exchange. And so what you end up with is a jumble. A total jumble. Big picture on this. Big picture on this. You might think you're passing on the chromosomes you got from your mom and your dad, right? You might think, oh, I can pass on chromosome 1, which I just erased, the one that I got from my biological mom. I can pass that on to my kids. Or I can pass on the chromosome 1 I got from my biological dad. And that's just not true. You can't. You can't because that chromosome doesn't exist anymore. It has been shuffled with the one you got from your other biological parent. And I mean that, like, it's been kind of in many ways randomized. So the alleles on each of your copies of chromosome 1 are a mismatch of the ones you had from each of your parental chromosomes. So that is a slightly more accurate version of this, right? A slightly more accurate version. Now, obviously, I'm not going to draw that out here. All right, so my apologies. There's only so much I can draw here. So if you thought, hey, I can make 8 million different sperm or 8 million different eggs, no, it's actually infinite, right? It is effectively infinite because it's not just 8 million chromosome combinations. It's the fact that each of those 8 million combinations has another multiplication to it. But I'm going to move on from that. Okay, so you might be looking at this and saying, all right, wait, aren't there 2 divisions of meiosis? Yeah, there are. Meiosis 2 is nothing special. Meiosis 2 is nothing special. In fact, meiosis 2 is basically identical to mitosis in that during meiosis 2, that's when you segregate or separate the sisters. All right, that's where you separate the sisters. So after meiosis 2, you end up with 4 cells. And obviously I'm gonna like run out of space here because this is what ends up happening. And I could have that. And you'll see, hopefully, the end result of this, why this is so important. Obviously, there should be nuclei here. I just don't have space to put that in. Okay, well, so what's the end result? Please notice, we have 4 cells, and they are not in any way genetically identical to this original parent cell. These 4 cells right here, which I'm going to haphazardly like convert into sperm. These 4 cells here have half the chromosomes. They have half the chromosome number. So while we started with a cell that was 2n equals 6, we now have cells that only have 3 chromosomes. We would designate that as n equals 3. Not 2n, because we don't have 2 of every chromosome. We only have 1, right? And if this were a human cell, we would have started with 2n equals 46, and we would end up with just n equals 23. Those sperm, or if these were eggs, could fuse with another gamete. 23 plus 23 equals 46. Quick recap. Remember, we've got 3 different parts here that lead to genetic diversity. The fact that you can generate new alleles, new versions of the primase gene, new version of the keratin gene, et cetera, through mutations. Right? That's where you get P17 as opposed to P14, right? Just the different little b and big B, if you want to use your like old school like notation. Right? There's crossing over between the actual homologs, which basically randomizes which alleles are on which chromosome. And then we segregate the homologs from each other in a random manner called independent assortment. All 3 of those steps, plus our 4th step, which isn't even shown here, which is, well, when you actually have like mating, which sperm and which egg meet to some extent is randomized, right? Right. There might only be, in a human, there might only be 1 or maybe 2 ova out there, but which of the sperm leads to fertilization to some extent is random. Is it this one? Is it this one? Is it this one? Is it this one? Or is it something else completely? So that'd be a 4th level of genetic diversity, because there's randomization in terms of which gametes actually fuse. Okay, that's the big picture here. Hopefully you've realized that meiosis is all about genetic diversity, cutting the chromosome number in half and generate unique cells that can lead to unique offspring, while mitosis, which was on the other pathway over here, just regenerates the parent cell. Which cells do which? Well, if most cell types permanently inactivate the meiotic processes genes, so most of these processes don't happen in your typical cell, but there are a certain subset of cells, namely cell types inside your ovaries or testes, oogonia or spermatogonia, that if they receive the appropriate hormonal signals, will undergo this process and result in gametes. Okay, that's it for today. One last thing. I can't believe I forgot this. The last thing I should say, despite the fact that this is already an overly long video, this right here is what's on a Punnett square. I need to mention this. This is what's on a Punnett square. So when I say it's on a Punnett square, I mean, you want to think of a Punnett square, which I know you've all done this before. You're like, right, capital B, little b, which is the typical way of drawing it, right? Or maybe it's a combination of alleles. Maybe it's like capital C, big W, capital C, little w, little c, big. Those are the combinations of alleles on these chromosomes. If you really wanted to be accurate about it, I'm going to draw you an accurate Punnett square, or a slightly more accurate Punnett square. A slightly more accurate Punnett square would be, well, here's a sperm. And in that sperm, in the nucleus, there's a chromosome that has a capital C on it, and a chromosome that has a capital W. On this sperm, there's a chromosome that has a little c, and a chromosome that has a big W. Another sperm, And these are the different combinations. So when you draw a Punnett square, what you're actually doing, I'm doing this very poorly with a not so fine point marker, um, is you're actually drawing out all the possible allele combinations that are on your gametes. And over here is where you draw your ova combinations. And then here, these are where you draw your zygotes. All right, folks. Hopefully that gave you an overview of how this stuff works to some extent. I'll take your questions during class time. Best of luck, everybody. I will see you guys soon. Bye.